I hope you can all hear me, and I hope you can all hear my voice. So let me put on the screen again, share your screen, and I'm gonna turn on this slideshow. So in our previous session that you must have, you know, I hope you have received the notes as well, and you have read from the notes and through the notes as well. But so what we did previously, we were doing screening techniques, and especially we were discussing that there are two broad categories. One is primary, and the other one is secondary screening of microorganisms. So in the previous session, we have looked into primary screening techniques, and now we're going to look into secondary screening of microorganisms. Primary screening helps in the detection and isolation of microorganisms from the natural substrates that can be used for industrial fermentation. So this is essentially what we've been doing for primary screening. But what we have also seen that primary screening only gives you an indication of a microorganism, which is useful. And it can give you a colony in hand um, that seems useful, right? But we do not have details. We do not have details. What is this microorganism? Or is it a new microorganism, new strain, or maybe an already existing genus? What kind of microorganism is there? And then we do not have much details about the production of the substance or the chemical compound we're after, the antibiotic, or um, any other product or yield of the microorganism we are after. So this secondary screening of microorganism is supposed to give you a detailed picture of the research you're going to carry on in order to identify the antibiotic, the minimum or inhibitory concentration, which the dosing, everything which you want to know, and everything you want to know about microorganism in detail with the research that you do. So let's look into the features and the factors and the points that can be covered as part of secondary screening, and they're numerous. And we're going to look into them step by step. We're going to see how many things can be useful, right? So, so the information that secondary screening provides, it actually covers these four broad aspects. That is ability or potentiality of the organism to produce metabolite that can be used as an industrial organism. Of course, oh, you want to see whether this microorganism is suitable for the production, and the entire research will go around to see whether the organism is safe and whether the organism is safe for food industries or any other industry. So you want to see suitability of the organism, right, to go in any kind of industrial process. Then you're also looking into quality of the yield product as well. So you do know that there is a chemical compound which is useful, and you do know that this is the compound we're after, but we do not know whether this organism gives you sufficient yield, and is it cost effective, is the quality enough to go and can be used pharmaceutically. There are details that you need to know, and then the type of fermentation process that is able to perform, right? So you need to understand more about the microorganism, what kind of media you need, what kind of nutrient condition that it needs, what kind of fermentation process needs to be devised, whether the work has to be done on screening, on upstream processes, or maybe it has to be done on extraction and the downstream processes. And fourth one is elimination of the organism which are not industrially important, right? So once you're trying to understand the ability and potentiality of this microorganism, and then you're trying to understand the quality of yield and the kind of fermentation. Once you work on these three, you know which microorganism are industrially important, and then you can eliminate the ones which are not industrially important, right? So to evaluate the true potential of the isolated microorganism, both qualitative and quantitative assessments need to be done. And sensitivity of the test organism towards the newly identified antibiotic needs to be done, and it has to be done quantitatively so that you have a much better picture, right? So what are the things that you are going to identify in a microorganism? What are that potential that you're looking for? So number one, it is to determine the product produced by organism is a new product or not. So so you need to work on this. You need to know whether you're working, for example, we're talking about antibiotic, and the kind of species that you identify and the kind of the chemical compound that species was producing. It was nothing but the same group of penicillin you've been working on with. So, but what you were after a new compound. So you have to do some research to understand whether it's something new that you're working on. Then a determination should be made about the yield potentiality of various isolated microorganisms that are detected by primary screening for that new compound, right? So now you have to look whether the microorganism can give you a sufficient yield, and if it can give you sufficient yield, then it is suitable for industrial processes. It should be determined, point number three, about the various requirements of microorganisms. Of course, we need to know the media requirements, the climatic environment, the climatic conditions, the environmental conditions, aeration, temperature, pH, all that is suitable for that microorganism. Then it should detect whether, point number four, they should detect whether the isolated organism is genetically stable or not. So sometimes you have to examine this point, whether they're genetically stable or not, and why. Because there are microorganisms that exchange their plasmids very often, they exchange their genes, and and there are microorganisms that mutate very often. So we need to know that genetically they're stable enough. So once you identify, and then you go for maintenance, subculturing, and maintenance of the stock microorganism, and then you're going to bring it back for fermentation process, industrial process again. It, you don't expect the organism to change, right? You want it to be genetically stable. So you also want to examine this factor as well. Point number five is it should reveal whether the isolated microorganism is able to destroy or alter chemically their own fermentative product. Now, this is something very critical as well. It happens that whatever product it's producing, after some time, it might also, over some time, it tries to break that compound down, right? So it might chemically damage the product that you were after. So sometimes it happens, it starts to, produce enzymes which accumulate in large quantities, and then it starts to degrade the product itself. Then it should reveal the suitability of the medium or its constituents, chemicals for the growth of microorganism and its yield potentiality, right? So whether you were working on the environmental conditions like pH, you should also be working on the suitability of the medium and the nutrients that are going in to have a better growth of microorganisms. Now, it should determine the chemical stability of the product as well, whether you get the product and it stays chemically stable or not, or maybe it deteriorates on its own, so you have to determine this factor. Now, it should reveal the physical properties of the product. But why physical property? Meaning what's the chemical structure? What is the boiling point? What is the melting point? What is all those physical properties that we used to study about any kind of chemicals? That needs to be determined, and all of that will be determined in secondary screening process. Now, it should determine whether the product produced by a microorganism in a fermentative process is toxic or not. Of course, it's so understood that if you have to use it in industries, uh, that that kind of product or any other byproduct which is being produced during that fermentative process, it shouldn't be toxic at all. So you have to do a very thorough screening of all the chemicals and the products which are being made. The point number 10 is that secondary screening should reveal that whether the product produced in fermentative process exists in more than one chemical form. Now, why is this important? Is that the amount of formation of each chemical formation of those additional products is particularly important since their recovery and sale as byproducts can create greatly improve the economic status of the fermentation industry. Now, the thing is, does it exist in more than one chemical form? Because sometimes there are byproducts which can also be of benefit to industries, and sometimes there are byproducts which can negatively affect the yield of the main product that you were after. So, in either of the case, you have to determine whether there is a second chemical form and that you can utilize that second chemical form. Because what you can do, you can chemically change one chemical form and get the yield, and it will actually double the yield, and it will also definitely improve the economical status of that industry, particularly those who own that industry, and the profits that is achieved over time. So the new organism should be identified to the species level. Of course, it is understood when you have the organism, you have to know what is the genus, what is the species, and also what strain of the microorganism that you're working on. You have to be very certain and particular. So it should select industrially important microorganism and discard others. This is the fourth point that we just discussed when we were discussing those four broad points. So of course, eliminate the ones which are not useful and keep the ones which are useful. So it should determine the economic status of fermentation process undertaken by employing new isolated microorganisms. Of course, at the end of the day, the industries and are going to look into the economical status, and they're going to look into the expenses and the overhead, and they want to determine a new microorganism and the higher yield of the product because they want the processes in their industry to be cheap so that the overall revenue or the profit that they generate is more. So, though you are researching and you are researching scientifically, but you're doing the scientific research to keep the economic status of the industry in mind, and because you want people to achieve more profit out of the new product or new organism that you have in hand. So what are the methods that you can use for secondary screening? So there are several techniques and procedures that can be employed for secondary screening. However, only a specific example or estimation of antibiotic substance produced by species of streptomycin will be discussed here. Now, why is this important is because secondary screening would add a lot of details, and in order to determine what kind of details we would be discussing, we are circling around just one kind of example, and that is about the antibiotic we are uh, uh, we are uh, we are expected to produce, and we are expected to produce it from a streptomyces strain, right? So whatever example we're going to study of secondary screening, we're going to study out of the context of streptomyces producing an antibiotic, right? So there's this one method called giant colony technique. What is this giant colony technique? In this technique, what you're doing that number one, you're going for those kind of antibiotics which diffuse through the solid medium. This is important because later on we're going to see and we're going to look into the kind of antibiotics which are not which cannot diffuse easily through a solid medium, and for that we need liquid medium. So we're going to look into those examples as well. But for now, we're looking into giant colony technique, which uses an agar as a solid medium and the ability of an antibiotic to diffuse. So what do we do in this this method that we isolate the Streptomyces culture, and it is inoculated into the central area of the Petri plate containing the nutrient agar and are selected. The plates are incubated until sufficient microbial growth take place, right? So let me draw like I've been drawing for previous examples. So let's go ahead and let's select a color and I will draw it for you. So what do we have here? We have a Petri plate. And in this Petri plate, we're gonna grow the colony, but we're only going to grow it in the center, right? So this is the center, and this is where we're trying to grow the colony, right? So, and which one it is? It is Streptomyces. And now what we're going to do in next step is that I'm reading point number 3 here, that cultures of the test organism whose antibiotic sensitivity needs to be measured. Now, that test organism, like we've seen in primary screening, that it is the known microorganism against which you want to test the antibiotic or any other property that you want to study, right? So let's say our test organism is an E. coli strain, right? So this E. coli strain is the test organism. All right, so what I'm writing is microorganism, MO for microorganism. So what we're going to do is take Streptomyces and we're going to streak from the edges of the plates up to the growth of Streptomyces, but not touching the Streptomyces. So it's almost like doing the streaking like this and then doing the streaking like this, but we do not have to cover the Streptomyces growth. So it's like streaking, go ahead, streak, 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 but leave the Streptomyces growth in the center. Once we do this, what should happen? It should happen that once you grow and streptomycin is producing the antibiotic it is producing, we should observe the zone of inhibition, but that zone of inhibition is very big and clear because there's one giant colony, and you can use this feature to assess the zone of inhibition and the distance, so that you can do some sort of quantitative analysis. So how will this zone of inhibition would appear? Zone of inhibition should appear something like this. So there is this big zone of inhibition, and this is your streaking of the test organism here. And this big zone, you already know about antibiotic sensitivity. So this big zone can be measured. Now let's look into what inhibition spectrum is. Inhibition spectrum is when you look into different test organisms, meaning you were testing it for E. coli. So let's say you also want to test against Staphylococcus species, right? And you also want to test this against, let's say, any antibiotic like Vibrio, and you also want to test it against, let's say, Salmonella, right? So when you're testing different microorganisms, and you have done the same procedure for this one, number one, for this one, for this one, and for this one. If you have done testing for different kind of test organism, so one, and you measured the zone of inhibition for each one of them, in the end what you're going to get, you are going to get an inhibition spectrum. So this is the quantitative assessment. This is in millimeters the zone of inhibition against this organism, against this, and this, and then eventually you produce for yourself an inhibition spectrum. Then there is another method known as filtration method. What is this filtration method? Is employed for those antibiotics which are poorly soluble in water and do not diffuse through the solid medium, right? So streptomyces, how do you do? How do you obtain that filtrate which would contain the antibiotic? So the following is the procedure that you follow to get that filtrate which can be tested. So streptomyces is grown in a broth and its mycelium is separated by filtration to get cultural filtrate. So you let streptomyces grow, you let it grow in a broth, and then you filter streptomyces, and what would you get in the end is a filtrate, but that filtrate is supposed to contain the antibiotic. Now various dilution of antibiotic filtrates can be done, and then you would add them to molten agar, and once you add them to molten agar, what would happen, that agar would solidify, but the agar would contain the antibiotic, right? So let me just explain things a little further, that what do you have? You have, let's say, a filtrate here. Filtrate, right? So this filtrate is added into this another flask, and this flask contains agar, but in molten state, right? So now you've added that filtrate in agar medium. Now this agar medium is liquid over here, and then what you're going to do, you're going to take a petri plate and you're going to start pouring your agar molten plus the filtrate that you've just added, right? And now this media will solidify, and this media would be containing an antibiotic that streptomyces was producing, right? Now what you're going to do later on, that you're going to take one of your test organism, and I hope the concept of test organism is clear now. So you're going to take one of the test organisms and you're going to do streaking. And what you're going to do, you're going to put lines of streak. So this is one organism. And then you're going to take another organism. So I'm taking another color. I'm choosing another color. So this is another organism, and you're going to put line of streaks. And then let's say you want yet another microorganism. So you're going to put another line of streaks. And then you can go ahead for another, and you can do like this. And what you're going to see, and what you want to observe here, is whether which of the test organisms, shows no growth over this agar. If let's say, let's be precise here, and let's use blue color to say that this was one, this was two, right? So this was three, and this was four, right? So what happens, that as you let the street plate to incubate, and after 24 hours, 48 hours, whatever the incubation period is, what you observe that uh, this streak number two that you added is not growing. All right, so sorry, both of them are gone. So let's say streak number one and streak number two are not growing. So what you can say in the end, you can say that test organism one, test organism one, and test organism two are sensitive to the antibiotic which the streptomycin is producing. So you're estimating now. And you're estimating the same thing you were doing with a giant colony method, but that was for solid medium diffusibility. But this one is the kind of is the kind of antibiotic which doesn't diffuse through the solid medium, and you have to prepare the filtrate and then assess the use of antibiotics. Okay. Other than the filtrate medium technique, there is another kind of technique that you can use and employ. That is liquid medium method. In this liquid medium method, you're also going to obtain the antibiotic in the liquid medium, and then you're going to use that antibiotic extract through another method or any other antibiotic assay method that you know, you can apply that crude extract there and you can determine the activities in a simple way. So what this method would do, in this method, you would take a conical flask and then you will add a very good nutritious media and then you're going to inoculate streptomyces and then you're going to incubate at room temperature. They are also aerated and they're shaken so that there's a very good growth of streptomyces. So you're going to let more and more growth take place. Now, keep in mind that liquid medium method is for further screening and to study the antibiotic in detail. So you're preparing a very crude extract. So you need a very large quantity of streptomyces to be produced. So what you're going to do is you're going to make sure that your nutritious media and all of those environmental conditions that streptomyces needs should be optimum and you should get a maximum growth. Then what you're going to do is take 5 ml or so from the broth and what you're going to do is centrifuge, and once you are doing centrifuge there's going to be a pellet formation and pellet formation is that growth of the microorganism and the cell mass of the microorganism, and it's going to settle down making a pellet. And what you're going to do is you're going to discard the pellet and save the supernatant. Why? Because the supernatant has the crude extract which can be used later on to do any kind of antibiotic activity, which is useful. Okay, now let's look into the importance of culture collection because I mean, what you're going to do after the primary and secondary screening. You've been doing this effort so that you can identify a microorganism which has an industrial use. So now what you're going to do, are you going to discard and you're going to let all the work go in waste, or you're going to maintain those microorganisms as culture collections? So of course you're going to collect all of these industrially important bacteria or yeast and fungi, whatever, which is good and important for fermentation and industrial use. So there are almost about 500 culture collections around the world. So you can simply tell them the genus, species, and the strain, and they can give you the species identified species and strain from their culture collections. So this is also very important. And what happens that industries sometimes they maintain their own culture collections of their own patented microorganism which they use particularly for the kind of fermentation they perform. Um, the prime function of a culture collection is to maintain the existing collection, of course. So you will maintain the kind of bacteria you have, and you will make sure that it doesn't, the identified microorganism is not lost. And then there are problems associated, of culture maintenance, like how you're going to let it stay contamination free and how you're going to preserve it for long. So, but there has been advancement in development, and there has been techniques like lyophilization, which is a cryopreservation and freeze drying technique. Both of them are very important so that you can do the collection in a miniaturized method, and it's just a powdered form of microorganism, and then you put that microorganism over the culture or the broth medium again, and the same growth will be there on uh, in the fresh media after incubation. So these culture collections are important. So use of microorganisms selected from a culture collection obviously provides a significant cost saving because you do not want to lose, you want to just simply go and collect the microorganism you need. And of course, why is there's a significant cost saving? There's a significant cost saving because environmental isolation that you have to do involves extensive research, and that is very time consuming, and also it utilizes research is expensive, so it utilizes a lot of raw material, which can be very expensive to perform on a very large scale. However, the disadvantage is that the competitors have access to the same microorganism. Of course, culture collections are important, but why is this a disadvantage? There is a disadvantage because if you are an a person who's running the industry, and you have this particular product, you want that particular product due to that microorganism to be confined to you and to your industry. That's why you want to patent different procedures, microorganisms, and the product that you can use it for your industry solely. But if you're using culture collections, which is a service available globally, nationally, and worldwide, then it becomes difficult for you to not maintain that monopoly because there are competitors around who have access to the same microorganism, and the kind of economical financial monopoly that you want to build up as an industrialist will not be possible. Okay, with this we come to an end of our isolation and screening techniques, which includes both primary and secondary techniques. Um, so I'm going to end today's meeting.